Is that okay? Hello?
Yes, madam.
Yes. Everyone agreed on that? So you have to get your attendance, okay? You have to talk, my dear.
Yes, madam.
Okay, right. Let's start then. Histology revision. I have selected several questions from areas covering over the last few years, and selected few areas which are very common to appear in the exam wise. So one thing I have to tell you before we start is that the essay paper in anatomy, that is the theory paper 2, there are 5 questions and all 5 questions have a histology component. So 5 areas of histology in your general histology as well as systemic histology is bound to be covered in your theory paper 2. So that means it's quite a lot of histology curriculum. At least it covers about 1/3 of the histology curriculum. So the best is for you all to be really thorough with the basic histology which you studied at the very first day. And then when the systems comes, you can like directly apply that knowledge over to the system. For example, if you get a question asking you to describe the histology of esophagus, so you can write about the epithelium and then the other layers. You only need to know that because everything else is in basic histology. So all in all, epithelia is something that you really need to thoroughly know, because all 5 questions will ask you to, in one way or another, to describe the epithelia depending on that location. Right. So we'll go around the epithelia at first. So these are roughly the areas I have chosen. Broadly about epithelia and specification of epithelia, junctional complexes, then the tissues, cells, about the yes, that's good. Yes. Yeah, you are just logging the way that your name appears in the register. Okay. And then the bone and the types of bones and about some glands and few from different systems, right? So talking about epithelia. So what are the basic or the unique features of epithelia? Can someone quickly tell few things? Then we have a comprehensive list. How do you define epithelia?
They are usually present on a basement membrane.
How do you define epithelia? Definition, if somebody asks what is epithelia, what would be your answer? Where do you find epithelia? That is the definition.
Usually,
They cover the surfaces of organs and tissues.
50% correct. You get 50% of the marks allocated to that question. Yes, it covers the body surfaces. And it does what? Does it only covers? It covers your body, yes. And then it goes inside and lines your body cavities and the tracts as well, right? Yes, because it's the endothelium, it's the mesothelium in your body cavities and innermost lining of all these GI tract, respiratory tract, and so on. So the definition, you need to have two keywords. Epithelia are the cells that either covers body surfaces or lines the body tracts and internal surfaces of the body. Right? So that's the complete answer. So you have to always think how to get the maximum marks by writing the brief answer. So epithelia either lines or covers the body surfaces. Right? Then let's quickly enumerate or list the unique features of epithelia. I heard somebody telling the correct answer before. So quickly tell one by one. One student can tell and we'll compile a thorough answer for that. Okay, unique features of epithelia. What's the? So you can add the first definition as the first point. The epithelia cells that either covers or lines the body surfaces. Yes, good. That's number one. What is number two? Start quickly.
Those cells lie on the basement membrane.
Very good. All these epithelia cells lies on a basement membrane. So that's a unique feature of epithelia. Okay. Number three.
They have free surface.
Can't hear. What is it? Unique features that, what are the features that it separates from all the other tissues? So it has a basement membrane, yes. All epithelia has a basement membrane. Just with the basement membrane, you can add up the other things because it has a basement membrane. Would that basement membrane permit the neurovascular bundles to pass through?
No.
No.
They receive the
So
nutrients by diffusion.
Yes, epithelia lacks blood vessels or nerves because there's a basement membrane and it won't allow the blood vessels and nerves to pass through. So it lacks blood vessels or nerves and gets its nutrition through simple diffusion. So that's the third point. What's the fourth one? That's the actually fourth point. What's the fifth one?
It has free
Between
surface.
cells. Sorry? Yes?
It has free surface, and between cells they have no space.
Very good. Can you improve on that answer? It's the meaning is correct. How do you say? It has a free surface. It has two lateral surfaces as well as it has a basal surface. We called epithelia are polarized cells. Yes, true. And you said it has what? No spaces between two cells. Yes, correct. What? How do you call these two spaces between cells? No, intercellular spaces. Yes, so that word needs to be there. That simply explains that you know. I always tell students that you know this stuff, the histology, the gross anatomy, or whatever, you know the content. Yes, there's some problem in getting it into writing within a limited time period. When you write, sometimes though you know it, you don't properly convey. So that's why you have to be very brief as possible as brief and learn to write the answer. So there's no intercellular spaces. If so, what is there? Now you said there's a free surface, there's two lateral surfaces and there's a basal surface. What is there between two lateral surfaces and the basal surfaces? Let's talk about those two surfaces. What is there? If there's no intercellular space, what is there? Another cell specialization is there. What's that? What's that? Anyone quickly try?
Cell junctions, madam?
Cell junctions, exactly. Another unique property of epithelia. Junctional complexes, all these tight junctions, gap junctions, desmosomes, hemidesmosomes exist between two cells or either between the basal cell and the basement membrane. So cell junctions are another unique feature. One more thing, another more unique feature. Think of in embryological terms.
They have ectodermal energy.
Yes. What? They derived from where? Where does epithelia? Sorry?
Derived from all three germ layers?
All three germ layers. They are derived from ectoderm, mesoderm, endoderm. Give me examples.
Ectoderm, epidermis of skin.
Yes, okay. Mesoderm?
Mesoderm, endoderm, lining of GI tract.
Yes, endoderm lining, yes. Mesoderm? How do you call them epithelia that is derived from mesoderm?
Mesothelia.
Mesothelia. Right, good. Now, how many unique properties we have discussed? 7 altogether, right? They are covering or lining the body surfaces, no intercellular spaces between the cells, all epithelia lies on a basement membrane. There are no blood vessels or nerves. There are cell junctions between them. They are derived from all three germ layers. And also another unique feature is all epithelia has either one layer or more than one layer, either depending on the type, simple or stratified. So if you get a question asking you to describe the basic features of epithelia or unique features of epithelia, you just have to write on those things. Is that clear?
Yes, madam.
Yes?
Yes.
Is that? Yes, okay. Right. Let's move on to the next question. State the classification of epithelia. How you classify epithelia? Now I want you to take 1 second, think and give me a full answer. Classification, describe the classification of epithelia. Commonly you get not just epithelia, any other thing. Classify bones, classify epithelia, classify this, classify that. You get a question most oftenly. So how you classify epithelia?
Classify
Yes.
according to whether they have membranous function or glandular. And then there is another category for simple and stratified.
Okay. 3. How many classifications? There are 3 classifications.
Shape.
Depending on the number of cell layers, are then saying simple or stratified. Yes. So first basis is number of cell layers. If there's one cell, you call it simple. More than one, stratified. Then the shape of the cell. Second classification is depending on the shape of the cells. Third classification is presence of surface specializations, depending on the surface specializations. So the classification number 1. So you have to write all these things when you are asked to describe the classification of epithelia. First you write there are 3 types of classification. Classification number 1, 2, 3. Number 1 depending on the number of cell layers, and you classify the epithelia into 3 categories, that is simple, stratified and pseudo-stratified. So you quickly write one sentence saying or drawing what they are. And classification number 2 is the depending on the shape of the cells. So could be squamous, cuboidal or columnar. Classification number 3 is presence of, now that's the surface of the epithelia. Depending on the surface specialization, you could go and describe even more than that. See, for example, whether the cilia are present or sometimes the microvilli are present. So that's basically the classification of epithelia. And then, what are the types of simple epithelia? Quickly tell me how many types of simple epithelia?
3.
What are those?
Simple squamous, simple cuboidal, simple columnar.
Still you could improve on that. Simple epithelia, I would say there's 4 types. What is the fourth type?
Simple columnar
Pseudostratum.
unciliated epithelium and simple columnar ciliated epithelium.
Yes, okay, that's good. Then I would say now 5 types. There's one more. What is that?
Pseudostratified columnar.
Pseudostratified columnar.
Pseudostratified, yes. So you have to tell that in first go. Right, okay. Now that's the types of simple epithelia, okay. So you can't forget that. Then give me functions of simple squamous epithelia. List few functions, 1 or 2. What is the function of simple squamous epithelia?
Protection.
Reabsorption.
Protection, absorption, yes, and filtration, yes. For example, in the kidneys, good. And give me examples. Where do you find the simple squamous epithelia? That you need to know by memory. Quickly, few sites.
Alveolar lining.
Alveolar lining, good.
Capillaries.
How do you call it?
Capillary endothelium.
Yes, if you just say capillaries, you won't get any mark. So you have to tell capillary endothelium. Okay, alveolar lining, capillary endothelium. Then
Bowman capsule.
others. Yes,
Bowman capsule.
Bowman's capsule. Very good. Where else? Where else? Quickly, quickly. What lines the peritoneum like that? Some pleura, lining of peritoneum, pleura. Do you get simple squamous epithelium?
Yes, in the form
Yes,
of mesothelium.
mesothelium. Yes, yes, mesothelium. So those are the places where you find the simple squamous epithelium. Where do you find simple cuboidal epithelium?
Glands.
Give it. Yes.
Glands.
Now don't, if you write just glands,
Pancreas.
not correct now. Specify a gland. Yes, it's present in so many glands, but specify a gland and
Pancreas.
where in the gland. Simple cuboidal epithelium.
Pancreas gland.
If you say
Thyroid gland.
glands, no, glands doesn't have cuboidal epithelium. Where in the glands?
Duct.
Ducts,
Pancreas
yes.
duct.
So, excretory ducts of the pancreas, yes. Then, where? Name another gland.
The
Thyroid gland.
Yes, parotid gland, excretory duct of parotid gland or ducts of the pancreas, like that. You have to give a complete answer. Then simple columnar epithelium. Where do you find? Quickly tell me the sites. Simple
Lining.
columnar.
Lining of stomach.
Lining of stomach, small intestines. Yes. Then few organs.
of
Name few
uterus.
organs. Oh, yes. And then gallbladder. All those places. So at least know 3 places. Simple columnar ciliated epithelia. Yeah.
Fallopian tube.
Fallopian tube, very good. It's found in the fallopian tube. Pseudostratified ciliated columnar epithelium.
Respiratory tract.
Now, respiratory tract won't be correct. Where in the respiratory tract? Name it.
Respiratory epithelium.
It's also known as respiratory epithelium, yes. But throughout the respiratory tract, you don't get this respiratory epithelium. For example, if you go to the alveoli, no, there's a simple squamous epithelium. If you go to the alveolar ducts, no, you don't get respiratory epithelium like that. So where in the respiratory? So that's why I say you have to specify the part of the respiratory tract.
Respiratory tract
Pharynx,
except alveoli.
larynx,
Sorry?
pharynx, larynx, and then
Yes,
trachea.
Trachea, yes. Trachea, primary bronchi, like that. So you have to specify like that. Right. Okay. That's basically about the simple epithelia. Next, I would ask you to describe what is cilia. Pseudostratified ciliated columnar. So what is this cilia? What is the structure? Describe the structure of a cilia. These are cilia, right? So you can see from the apical surface of the cell, you get these very thin hair-like projections known as cilia. What are they? And you can visualize it under the light microscope. What is the, if you take a cross section of cilia, what would you see?
Microtubules.
Microtubules, very good. And they are arranged in a very unique manner. What is, how do you describe that arrangement?
9 plus.
Doublet
9
forms.
plus 2, yes, 9 plus 2 arrangement. So you can't forget any of these things because you get a simple question like describe the histological structure of cilia and you get 20 marks. So that's a big amount and very easy to score. So you have to write all these things and learn to draw a cross section as well. So arrangement of microtubules is very unique. And how does cilia differs from microvilli? What is microvilli? Does it also have this 9 plus 2 microtubule arrangement?
No, madam, they are projections of the plasma membrane.
Very good, very good. Finger-like projections of the cell plasma membrane are known as microvilli. So they lack these cytoskeletal elements like microtubules. And give few things. What if you get a question to ask you to describe microvilli, what else would you add up? When on a surface they are present, how do you call that surface?
Absorptive surface.
Sorry?
Brush border.
Brush border, yes. Brush border or striated border because they are like a brush, very thick and you can see on the surface of a cell. So you call it brush border. And where do you see this brush border? Like this now. In small intestines, yes. Surface projections, and there are, if you want to add a couple of more details, there are tight intercellular junctions between the cells there, and because cell junctions are there because of the cytoplasmic extensions to give the strength to the cell, and these are the microvilli. Well, what is the purpose of having microvilli?
Increase the absorptive surface.
Increase the surface area for absorption. Good. Then, okay, quickly list what are the types of stratified. Now, all this is about simple epithelia. What are the types of stratified epithelia?
stratified squamous, stratified cuboid, stratified columnar,
Only 3? I would say there's 4 stratified.
Transitional epithelium.
Transitional epithelia. So you can't take time. You have to quickly tell.
Transitional epithelium.
Yes, transitional epithelia is the 4th type. And how you name these epithelia? Like if you take stratified squamous epithelium, there's a basement membrane and there are more than one cell layer. For example, here, stratified squamous epithelium. Look at this picture here. So there's a basement membrane and there's several cell layers. So if you look at the basal cell layer here, the cell layer that is sitting on the basement membrane, what shape is that? Can you see a squamous shape there?
Cuboid.
Cuboidal. And when you move in, the intermediate cells are with many shapes. We call it polygonal shapes. And the surface cells are scale-like and squamous. Look at the stratified columnar epithelium. There's a basement membrane and basal cell layer is again cuboidal. Middle cell layer pretty much polygonal or cuboidal. And the surface cell layer is columnar. So what gives this stratified epithelium its name? Because it has many cell layers, the surface cell layer defines the stratified epithelia, right? So we name the stratified epithelia. Now you could name this, if you name this stratified epithelia depending on the basal cell layer, we can call this stratified cuboidal or stratified squamous. So, sorry, stratified columnar. We call this stratified columnar because the uppermost cell layer, the shape is the columnar. So that is also important for you to remember. And stratified squamous epithelium. What are the types of stratified squamous?
Stratified squamous keratinized epithelium and stratified squamous non-keratinized epithelium.
Yes, keratinized and non-keratinized epithelium. Non-keratinized epithelium, where do you find?
In
On the wet surfaces.
Where?
Lining wet surfaces.
Lining?
Wet surface.
No, you have to specify where. Tell exactly.
vagina,
Yes.
oral
cavity.
mucosa,
Oral cavity. Yes. Then going down. Pharynx, esophagus, anal canal. All these places you get stratified squamous non-keratinized epithelia. And tell me a few other points about this epithelia. What are the typical features of stratified squamous non-keratinized epithelia? This is, what would you see? Where the cell production occurs, because there's more than one cell layer, where these cells are produced.
from the basal layer,
Basal layer. So as a result, what would you see in the basal cell layer? Histologically?
Meiotic.
Mitotic. Meiosis doesn't occur, right? Mitosis, because these cells divide by mitosis. Meiosis occurs only in one place. Where?
Germ cell.
germ cell layer.
Yeah, either spermatozoa or oocytes in the reproductive tract only. Therefore, mitotic cell bodies are present. That's again unique feature, and another unique feature. So the bottom cell layer keep on dividing and adding cells upwards. And then what happens to these surface cells?
They have to be flattened.
Yes, they get flattened, scale-like, and sometimes get out of the surface. Then stratified squamous keratinized, where do you find?
dry areas like skin,
Skin, yes.
hair,
Hair. Okay.
nails,
Nails, yes. Stratified squamous, keratinized. Okay. As that then stratified cuboidal.
Slit
Loud. Where do you find stratified cuboidal?
glands?
Now, if I told you again, if you write sweat glands, you won't get any marks. Where in the sweat gland? Because just get this straight into your head. The glands. What is a gland made up of? What are the two components of a gland?
Ducts and alveoli.
the secretory unit and the excretory unit.
Yes. Yes. Glands has a parenchyma and stroma. In other words, secretory portion of the gland is known as glandular parenchyma, and the stroma, that is the connective tissue portion, that the capsule, which it divides into lobes, lobules, all these things, right? So then if you think of the stroma, that is mostly connective tissue, collagen and fibroblast, blah, blah. So if it's the glandular parenchyma, then the actual glandular cells and alveoli. So, and the ducts, yes. So where is this stratified cuboidal epithelium you find? Where in the sweat glands?
Secreted duct.
Ducts of the sweat glands,
Larger
ducts of the
ducts.
pancreas, ducts of the parotid, yes. So that's the stratified cuboidal epithelium. Stratified column epithelium, one place. Very rare, but you need to know where is it. Stratified column.
Conjunctiva of eye.
Yes, ocular conjunctiva. Yes. Then transitional epithelium. Now correctly tell where. Transitional.
Bladder lining.
Bladder lining, not just bladders. Keep on listing.
Ureters and urethra.
Bladder, ureter, urethra, prostatic urethra, and up where in the kidney? Which part of the kidney? Renal, which opens
Renal
into
pelvis.
pelvis.
Renal pelvis.
Renal pelvis. Starting from there, you get transitional epithelium. Now tell me the key features of transitional epithelium. Why it's called transitional? First of all, why it's called transitional epithelium?
Because morphological number of cell layers can be changed during
What's the other thing? Number of cell layers can be changed. Is that biologically possible?
no madam,
No.
morphological.
Shape of cells. Shape of cells.
Yes. Shape of cells can be changed. Number of cell layers won't be changed.
Constant.
Yes. So why it's called transitional epithelium? What it transits between?
During stretching and relaxation between that function.
It's called transitional epithelium because the cells, the shape of the cells are transitional between cuboidal and squamous epithelia. It's a transitional form. So, if so, what are these cell layers? How you call these cell layers? The cell layer lying on the basement membrane is basal cell layer. And what's the shape of that cell layer?
Cuboidal.
Cuboidal cell layer. Cells lying on the basement membrane. You can see here the cuboidal cell layer. Intermediate cells, what shape are those? Polygonal shape. And the surface cells are large, rounded, and sometimes all these cells are single nucleated. Surface cells may be binucleated. And it also has a scalloped surface outline. So those are the unique features of transitional epithelia. So once again, list the unique feature. Transitional epithelia comes again and again and again in questions, like 1,000 times. So you can't escape from that. What are the features? List them quickly. No answer?
They have more than 4 to 5 layers.
Okay,
They
good.
have and they have 3 layers changing morphological shapes like
Yeah.
basal layer cuboidal, intermediate layer polygonal. Apical will have large rounded cells, either uninucleated or binucleated.
Yes.
Uh,
And surface is scalloped. You know what is a scallop,
Scallop.
right? Scalloped surface outlines. Yes. Okay. At least that. And if you want to add more, this surface has a very thick lining, very dark, thick cellular coating, so that it helps to resist the toxicity of urine, because the urine is heavily concentrated and very toxic substance that the cells might get damaged by its toxicity. To avoid that, there's this dark, dense, thick coating in the apical surface. Right. What are the functions of transitional epithelia? That's again important. Functions. So as I said, withstand. It can tolerate the toxicity of urine is number 1. Okay. Right. Then what are the functions? What does it
It can
allow?
expand.
Yes, it accommodates great degree of stretching. It can expand. Yes. Thirdly, because the urine is highly concentrated, what's its third function?
Resist osmotic.
Sorry? It prevents water getting absorbed from the cells into the urine because of its high concentration. So all these functions are performed by this transitional epithelia. You need to know for all epithelia, you need to know the function and how these cells are adapted to perform these functions and their location. So if you followed the class up to now, you can make a nice table. One column, the type of epithelia. Second column, unique features. Third column, special features to perform its function. Fourth column, where they occur. And that's it, right? So you make a table today itself, and which you can summarize the entire epithelia, which is very important. Right. Then quickly about, because we talked about cell junctions, quickly about the 4 types of cell junctions that you get. Those are the tight junctions. Then you get adherence junctions. Then you get gap junctions. There are large gaps or spaces between those cells, as well as the desmosomes, or if only one side of these desmosomes present, you call it hemidesmosomes. Again, you need to know a little bit of cell junctions. There's separate chapter in the Vitas allocated for cell junctions. You need to know where they occur and what is the unique features and what's its functions, that they add to the physical strength, and also these cell junctions, the cells are live components. So between the cells, the information and metabolites are thought to be exchanged via these cell junctions. So that's why we need to know a little bit about cell junctions. Okay. What's our next question is describe the cells, right? Describe the cells, fibroblast. Okay. Even the last question, like this 41st repeat exam, has a question asking you to which needed to describe the fibroblast. Okay. Tell the unique features of fibroblast. Okay, 1. Shall I disturb you? There's 114 students logged in, but only 99 has entered their names. So others please also enter their names and take a screenshot of that. Your attendance will be counted, that's why. Fibroblast, what are those cells?
They have irregular shape.
No, you can't say irregular. What's that? What? They have specific shape. Okay, irregular, okay. Irregular, yes. What does it have? Irregular shape, okay. What is a fibroblast? What is a fibrocyte? This is a very first stuff. You need to know very well now this. What is a fibroblast? What is a fibrocyte?
Fibroblasts have the ability to produce fibers and matrix of the connective tissue.
Fibroblast is an immature cell. Fibrocyte is a mature cell. So fibroblast is the active cell. Fibrocyte is an inactive cell. So that's the shape of the fibroblast with multiple cytoplasmic extensions. You can call cell processors, multiple cytoplasmic extensions. So fibrocyte is an inactive cell. Fibroblast is a very active cell. As a result, you can see all these intracellular organelles are very high in function. Right? What is the main function of fibroblast? So you should have revised this now last in March. Have you all seen the exam paper? Yes? Have you all gone through the paper? Essay paper? In the repeat exam? Hello? Yes or no?
No, madam. Not yet.
Sorry?
Not yet, madam.
Oh, yes, okay. So today, if you have access, probably you don't have access, probably get at least last 5 years exam papers, and you should now organize them into histology sections and different sections, and there's no need for you to write answers as such, but just go through them while you are studying, so you'll pay a lot of attention to that areas. So fibroblasts are responsible for these tissue damages, repairing tissue damage and synthesis of collagen fibers, all those active cellular mechanisms. You can't see fibrocytes or fibroblasts under light microscope. You will only see their flattened nuclei in hematoxylin and eosin stained sections. In the mature form, because they are inactive cells, cytoplasm does not contain many cellular organelles, and they are capable of this amoeboid movement. So those are the main features of fibroblast and fibrocytes. Okay. Fat cells. Second cell type is adipocyte or fat cells. Everybody knows it. Quickly list the features. Adipocytes. How you describe it? These are all connective tissue cells. Now we've covered epithelia, the first in basic histology, the second is connective tissue, right? So I have chosen few areas from each of those sections. This is connective tissue. So adipocytes are fat cells. Describe quickly.
those cells, they consist of a central large lipid droplet and
Yes.
peripheral cytoplasm.
Yes, a thin rim of peripheral cytoplasm and a large globular fat. Yes, very good. Nucleus? Where's the nucleus?
Displaced to periphery.
Yes, flattened, squashed and displaced to a periphery, and therefore you give what? What
Sorry, madam,
then
couldn't
you give?
understand.
Sorry?
Couldn't understand the question, madam, the last one.
The question is describe the histological features of adipocytes. We said it's there's a large globule of fat. Yeah, I'll put the slide. Now that's an adipocyte, right? You look at the cell, large globule of fat here, there. And where's the cytoplasm? The cytoplasm is pushed to a thin rim at the periphery because the fat globule occupies the entire cell here. The cytoplasm is restricted to a very thin peripheral rim. And the nucleus is also squashed, flattened, and located at the periphery of the cell, right? Now, what is this appearance reminds you of?
Signet ring.
Signet ring. Here's the signet, the nucleus, and it's a ring. Thus, it gives a signet ring appearance. What happens in H&E sections? Can you see this appearance?
No.
No, you see some empty space here because when you use alcohol for H&E staining, it dissolves the fat. So all the fat is being dissolved away. What you could see is this thin rim of cytoplasm stained pink in color and the nucleus only. So you just see some empty spaces with flattened nuclei. If you want to visualize the adipose cells like this, what are the stains you can use? There are special stains. We study of those special stains. What are those? You could add that as well, so you could get more marks. What are the special stains to stain lipids? Come on, at least one.
Biuret.
Sorry? You need to read quite a bit more of the Witters. What are the special stains? At least one student? Sudan stains, at least remember that. It's easy to remember. Sudans are special stains for lipids, like that. There's plenty of special stains for lipids, and one is Sudan stain. So you have to osmic stains, which stains there are in the Witters. They give some different slides as well with different special stains. So just have a look at them. Right. Let's move on to the next question. Plasma cell. Again and again. This has also comes in the exam papers quite a lot of times. Plasma cell. What is the histological features? What is its shape?
They are mostly spherical, circular shape. No, oval shape.
Oval shape, exactly. The unique feature is they are oval shaped cells.
Basophilic cytoplasm.
like the spokes of a wheel or like a clock face appearance. So that unique nuclear feature is called cartwheel appearance or clock face appearance. The cytoplasm is intensely basophilic due to rough endoplasmic reticulum, and their function, everybody knows, is the secretion of antibodies, right? So plasma cells. Next is mast cells. Now, okay, I have described three cells altogether. So you describe the mast cell. At least tell me the key histological feature.
Round cells.
Sorry?
Round cells.
Okay. Round or oval cells. Okay.
Oval nucleus.
Mhm.
Histamine and heparin.
Yes. Whereas it's one of its functions is secretion of histamine or heparin, anticoagulant heparin, yes, or histamine. Where are those substances in the mast cell?
In the cytoplasm.
In the cytoplasmic granules. Cytoplasm is packed with granules which contain either histamine or heparin. Very good. Then there's another unique feature. What is the most key feature? Most important key feature is the, it shows the property of metachromasia. Remember? Metachromasia is that when certain dyes such as the toluidine blue, when we use toluidine blue to stain the mast cells, these stains bind to this cytoplasmic granules and change its color to red. That's why you see mast cells as reddish in color. This property is known as metachromasia. So that is also very important property of mast cells. Right. Then now, is that okay? How you find? Is it too fast? What?
It's good, madam.
Sorry?
It's good, madam.
Okay.
Madam, we
Yes.
couldn't see the screen, madam.
Now what about now? And
Still not, madam.
still not. Okay. Let me see. Now?
No, madam.
No.
Madam, actually, what something happened when you were describing plasma cells. Part
Plasma
of it
cells.
was
Okay,
also not heard by us.
I'll put share screen again. Okay. Can you see now?
Yes, madam.
Okay. Let's go like that. You want to see plasma cells? Okay. Right. The plasma cells. Okay. So you have to remember this. Can you see now?
Yes, madam.
Yes, madam. Yes,
Okay. You have to see that it's oval shape, and on one corner of the cell you have this very rounded nucleus, and the nucleus gives its unique feature by nuclear chromatin arrangement, which we describe as clock face or cartwheel appearance, and the cytoplasm with H&E stains very darkly basophilic, because obviously it produces antibodies and therefore it has very abundance of rough endoplasmic reticulum. So that's quickly about the plasma cells. And then mast cells are round cells heavily packed with the cytoplasmic granules. The entire cell has these small granules which either contains heparin, and which is an anticoagulant, or histamine involved with these allergic reactions. and it shows the property of metachromasia, meaning that the basic dyes such as toluidine blue, which we use to stain the mast cells, they bind to these granules, and these granules are capable of changing the color of the toluidine blue to a deep red color, and that property is known as metachromasia. Right. Okay, now let's quickly move on to the bones. Now the bone cells are mainly of three types. Those are osteoblast, osteocytes, and osteoclast. Tell me the features of osteoblast. Quickly. Describe osteoblast.
They are present closer to the periosteum.
Yes.
They produce the matrix as well as they differentiate into osteocytes.
Okay. So the first thing, remember the fibroblast and fibrocytes, same principle you can apply here.
Osteoblasts are active cells.
What cells? Osteoblasts are?
Immature cells.
Immature cells, therefore they are active or inactive?
Active.
Active cells, right. So if active, where are they? Where would they be occurring in? Found in the periosteum as well as inside in the endosteum. So what is their function?
Secreting bone matrix and
yes, secretes the bone collagen and organic parts of the bone matrix, and they are being, when mature, they are transformed into osteocytes. Right. So that is osteoblast. And osteocytes, they are, of course, you can start with mature bone cells because osteoblasts get matured and become osteocytes. Now, during this maturation process, osteocytes acquire several different things, so structures surrounding it compared to the osteoblast. What is the structure of an osteocyte?
have a say.
Lacuna.
Sorry?
Okay. Have a say and
No, we are not talking about Havassian systems. We are just
Lacuna.
talking about... Sorry?
Lacuna.
Your question is, describe the histological features of osteocytes.
Madam, they are present within lacuna.
Very good. What is a lacuna? They are present within spaces known as lacuna. Good. Right. Very good.
They are like cages, madam. They are arranged around the lamina, which is surrounding the particular Havassian system.
No. Now we are going out of the track. They could arrange in the lamina, or if it's a trabecular bone, they are not arranged in lamina, right? So in spongy bone, they are not arranged in an organic system. So we are not going into that detail. We are just talking about the osteocytes. So they are the mature bone cells. They are derived by maturing of osteoblast. And now you said they are trapped within a space known as lacuna, and they occupy the entire space of the lacuna. Yes, very correct. And then what else? What is the shape of the cell? That's why you have to be very precise. You can write a lot of outside things, you won't get any marks for those.
skeleton.
What is the shape of the cell? There's lacuna, and what else the lacuna has?
Canaliculi.
Canalicula. Lacuna has small hair-like spaces, extensions known as canaliculi. Which part of the cell goes in this canaliculi? What does canaliculi employ?
Cytoplasmic processes.
Cytoplasmic extensions of the osteocytes. So the cell shape is the cell has lot of cytoplasmic extensions which are accommodated in this canaliculi, which are extensions of the lacunar space. Now, the cell has lot of cytoplasmic extensions. Cell is located within the lacuna to occupy this cytoplasmic processes of the cells. The lacuna has canaliculi. And these cytoplasmic extensions through this passing through this canaliculi, where would they go in? Where would these canaliculi join, these extensions? They join with the other canaliculi of other osteocytes somewhere here, and they join there, or they join with the central canal of the bone, which is the Haversian canal. Why? Why these canaliculi joins with other canaliculi? For what purpose?
Gain nutrients and oxygen.
For to get their nutrients, because the bone lacks blood vessels or nerves, so they communicate through this canaliculi, right? So you could add that as well. So that's how you describe the osteocytes. Okay. Third, what is the third cell type? Is the osteoclast, that I guess suppose everybody knows. Describe osteoclast. How many nuclei in osteoclast?
Multinucleated.
Multinucleated cells. And what color is the cytoplasm? Just like this bluish cytoplasm, basic cytoplasm like here. No, strong acidophilic cytoplasm. So it basically appears pink in color. Where would you see osteoclast? What is the function of osteoclast?
Bone resorption.
Bone resorption, or they are bone eating cells. They resorb the bone, or very helpful in repair process or bone remodeling processes, all these activities. And unique feature of osteoclast, once again, is when this, now this, suppose osteoclast is like that, irregular cell surface with multinucleated cell, strong acidophilic stains, and now that's a schematic diagram to show. Now this is the bone surface, let's say in the osteum here. The plasma membrane, which is in contact with the bone, is thrown up into a ruffled board. You can see it. It's a not nice smooth surface. It's been ruffled surface here. And this, between this ruffled surface of the osteoclast and the bone surface, you get some space. This is some greenish in color. Can you see the color?
Yes, ma'am.
Yes, this space, there's a specific name for this space. What is it?
Lacunae.
Yes, a lacunae. Yes, which lacunae? There's a nice name for that. Lacunae.
Horseships.
Houseships lacunae. Yes, that's the houseships lacunae, and the ruffled border, multinucleated cell, strong acidophilic stain, and the function is the bone resorption, or we call it in later bone eating cells. So all these facts you need to write under osteoclast at least, right? Okay. Then your most favorite question, which everybody would write, we'll just quickly go through. Describe the arrangement of compact bone. Yes. So what are the parts? Quickly tell. Compact bone. So, okay, that's the arrangement. What would you label here? Tell 1, 2, 3, 4, 5, 6, 7. Tell what are those? What would you draw or label? Okay, list the components of a compact bone. What would you list?
Haversian canal.
Okay, good. They have a Haversian canal, number 1. Okay, 2.
Madam, the lamellae.
They have lamellae. Okay, what are the types of lamellae? At this stage, you just can't say lamellae. How is lamellae? Okay, let's leave it like that. Okay. Haversian canal, you have lamellae. What else?
Osteocytes in lacunae.
Osteocytes, lacuna. Yes, then canaliculi. What else? You are looking at the diagram. So what else? What is this?
Walkman canal.
Sorry?
Walkman.
Walkman canals joining to Haversian canals. Yes. And the lamellae. Coming back to the lamellae, you said just lamellae. What are the types of lamellae? You just also have to label types of lamellae.
The outer and the inner circumferential lamellae.
Circumferential lamellae like that. The lamellae that goes around the Haversian canal, that is circumferential lamellae. Yes. Then what is the other type? What is the other type of lamellae? Looking at this diagram, you can't tell. Now here's one circumferential lamellae here that is arranged around the Haversian canal, and here's another one. You can see a small place here, another one around here, and in between them, there's also parts of lamellae, some interrupted lamellae there. How you call them? Interstitial lamellae. So there are two types of lamellae. Have circumferential lamellae, and in between the circumferential lamellae, you get interstitial lamellae. This entire arrangement of compact bone is known as what? Hello?
Haversian system.
Sorry? How do you call this structure? Osteon, right? This is an osteon. So if you get a question asking you to draw the structure of an osteon and describe, this is what you are supposed to do. Is it too difficult, too fast, or you are fed up? What? Shall I continue? Hello? Are you there? Can you hear me?
Yes, madam.
Then spongy bone. So that's compact bone. No?
Yes, madam, we can hear you.
Okay. Spongy bone. What's the difference?
They don't have osteons.
Are there osteons?
No.
No, there aren't any osteons. Where do you find spongy bone? In the bone marrow?
The ends of the
Ends of the long bones, yes. And in the bone marrow, you can see spongy bone. So small trabecules surrounding the red bone marrow, you can see spongy bone. So it's irregularly arranged lamellae, and there are osteocytes here connected by canaliculum, but no osteons as such. So for spongy bone, you can describe the osteocytes and lamellae, which are irregularly arranged, and osteocytes have canaliculum. That's all about spongy bone. Then the epiphyseal growth plate, again the last year exam in 2020 second MB, you got a question. If you have gone through it, describe the bone growth, the histological arrangement of epiphyseal growth plate. So just you describe the zones, all these 6 zones, zone of risk. So you have to draw this diagram and describe each of these cell layers, the reserve cell layer, the zone of proliferation, layer of hypertrophy, then calcification and osteogenesis, osteogenic zone, and the different qualities of these cell layers, right? Right. Next. Histological arrangement of a gland. Let's take the easiest gland, the thyroid gland. Describe. Histological arrangement of thyroid gland.
It is consisted of thyroid follicles.
Consist of thyroid follicles, yes. Very good.
Follicular cells line the follicle while the colloid is inside the follicle.
Now tell me about these follicular cells. What cells are those? The shape of the cells, what type of cells?
In active cells, the shape of the cells are cuboidal.
Simple cuboidal cells. Yes.
In inactive cells, that is of
Okay. What is the other cell types found in the thyroid gland?
parafollicular C cells.
Yes. Where would they be found?
Attached to the basement membrane.
In between the follicles, C cells. Why it's called C cells? C cells?
It secretes calcium.
It is present in the can be within the lining itself.
Sorry?
The parafollicular cells, they can be within the space between the follicles, or else they can be along the lining of the follicle.
Yes. Why are they called C cells?
They secrete calcitonin.
Where is it derived from?
Third pharyngeal pouch. No.
C cells?
Neural crystals.
C cells? Third pharyngeal. The answers are third pharyngeal arch, neural crest cells. What else?
They both have contribution to C-cells.
What? Okay, go and read your embryology and find where the parafollicular cells originate from. Right. Yes. Here. What is the structure? Can you see the screen?
Yes, madam. Ureter.
Okay, ureter. Now list the reasons.
And because of the center lumen shape.
Sorry?
I told it ureter with the shape of the center.
Okay, yes.
The lumen.
Lumen, okay, shape of the lumen. Then the mucosa. Reasons. So there could be more than one reason. Okay, shape of the lumen, the star shape, yes. The mucosa is what?
Transitional
Transitional.
epithelium.
And yes, what about the lamina propria? Very large area is occupied by lamina propria. And then you have this muscularis layer, longitudinal and circular. That's why you call it ureter. Those are some reasons. And then the adventitial layer. Okay. Identify the structure.
Esophagus.
Esophagus. So list reasons 1, 2, 3, 4, 5.
Epithelium is stratified epithelium.
Complete answers. Now we have covered the surrounding knowledge. So give complete answers. Epithelium is?
Epithelium. Stratified squamous non-keratinized epithelium.
Okay.
And it is presence of a submucosa also.
No, go in order. Does it has a lamina propria?
Yes.
Yes.
Yes. Okay. Then what?
Muscularis mucosa.
Very good. Then it has a muscularis mucosa. That's why it's thrown up into these folds. Yes. Then you can go to the submucosa. What is the special feature of submucosa? You can see it here. Submucosa has what?
They have
Glands.
Glands. What glands? If you just say glands, could be serous gland, could be what, any type of glands.
Mucus.
What glands are there in the submucosa?
Mucus gland.
Because
Mucus glands in the submucosa. Okay. Then what is the next layer?
muscularis propria.
Muscularis externa. What type of muscles are there?
Circular.
Skeletal muscles. Yes. Then what's the last layer?
adventitious.
What is this?
Adventitia.
Adventitia. Yes, adventitial layer. So those are the regions. Okay. I'll ask a question outside this, but relevant to the esophagus as well. You said in the submucosa you have mucus glands, right? Agreed?
Yes.
Yes. Tell me how you differentiate a mucus gland from serous gland.
Mucus glands are
Now all this time you have studied mucus glands and you have studied serous glands and you can identify mucus glands, you can identify serous glands. Tell me the features that help you to differentiate mucus gland from a serous gland.
Madam, the first thing is the stain thing, because the serous glands will darkly stain, whereas the mucus glands will stain pale. And
Yes.
also mucus glands, you can see the pale stain also, it will be like they are all lumen, but they are not lumen. Like when you see, you can see like gaps within the cell, but they are not gaps, they are the content.
1 minute. Now you said it looks like a lumen, but there's no lumen.
No, madam, not lumen, madam. It's like the content, it appears like there is nothing in it.
So you what's the key word? How you describe that appearance? There's a special word. You are very correct. You are describing it correctly. How you describe? You don't need lot of explanation. You use one term to describe its appearance. The cytoplasm is foamy in appearance, right? F-O-A-M-Y, foamy appearance. So everybody understand what is a foamy appearance, right? Okay. Cytoplasm is the glandular cytoplasm of mucous cells looks foamy in appearance. Some sort of cloudy, right? Foamy. Okay, right. What else? Good. Staining properties, foamy cytoplasm. What else? Why all of a sudden you're gone to silence? Yes. We have few more minutes remaining, so quickly tell the other features. What gives rise to, what is the substance that gives rise to this foamy appearance?
It will be more centrally located or else towards the apex.
What? What is centrally located? Sorry, I didn't hear what you said.
In serous cells, the nucleus is more towards the apex.
Can't be towards the apex. I'm yet to find a cell whose nucleus is towards the apex.
Madam, sorry, madam. In serous cells, the nucleus is not towards the apex. They are like most, not exactly centrally located.
Yes, yes, good. Right. But here, describe with them in mucous cells, where's the nucleus? Now, this foamy appearance is given by what is the substance inside this cell? Huh? Now, the mucous cells has this cloudy or foamy appearance due to this accumulation of mucus. Mucus is very thick and frothy. That's why it gives this cloudy appearance. So if mucus is a heavy, frothy thing, would you expect a nice rounded circular nucleus? The weight of this foam or the mucus is pushing the nucleus towards the basement membrane. So you would see a squashed, flattened nucleus close to the basement membrane. Agree? Yes or no? Hello?
Yes, ma'am.
Okay. So those are the three key features how you would differentiate a mucus gland from a serous gland. Right. Then, okay. Similarly, tell me the features of serous. You seem, why you are quiet? Either you don't understand. Did you get the thing? Did you don't understand, or you can't forget, or what? Is it too simple? No answers? You want me to stop now?
No.
So I'll just take one more thing and stop, right? Cardiac muscles. Describe the features. So we touched everything once we touch a muscle as well. Cardiac muscles. What is
Yes,
the,
striated muscles.
sorry?
Striated muscles.
Yeah, they are striated muscle. What the cell looks like.
Branch-like appearance.
Long cylindrical cells
Long cylindrical.
and branched. Yes, okay. Where's the,
Nucleated or wide.
yes, where's the one or two nucleus and where's the nucleus located?
Surface.
Sorry?
Middle.
In the center. Yes,
Center.
like in the skeletal muscle, it's the periphery. Now yes,
Surface.
it's in the center. And what is the other key feature? What are these things? Yeah.
It's branched.
What are these things?
Intercalated.
Intercalated disc. Yes. What is an intercalated disc?
Junctions. Cell junctions.
Gap junctions.
Yes. It's a specialized intercellular junction, and also it's the point of anchorage points for the cardiac muscles are attached there. So they transmit forces and the stimuli throughout the cardiac muscle. Therefore, the cardiac muscles can rhythmically contract and act like a syncytium. So that's the function of intercalated disc. To help cardiac muscles to act like a syncytium to contract simultaneously. Okay. What unique features are there in cardiac muscles other than that?
They are branched.
Yes, branched, yes. What are these cells?
Purkinje.
Purkinje fibers. So you have to write that as well. What are they, Purkinje cells? Where are they located?
Specialized cardiac muscle.
Specialized cardiac muscle cells. They are subendocardial locations, so they lie beneath the endocardium, right? Subendocardial location, you can clearly say. They are large cells and they have paler cytoplasm compared to the cardiac muscle cells. And the number of muscle fibers or myofibers are less than the number of muscle fibers that are present in cardiac muscle cells. And they have cell contacts with the cardiac muscle cells via gap junctions. Therefore, they enhances the electrical conduction. So that's about cardiac muscle cells. Anything? Right. And any other special features of cardiac muscle cells?
Rhythmically contract.
Rhythmically contracts, yes.
Involuntary.
What else? Okay. And about, just to summarize all three muscle types and to differentiate each of them. So we have talked only about the cardiac muscles. And if we are looking at all three muscles, their shape is long, cylindrical, skeletal, and cardiac, both muscles. And smooth muscles are spindle shaped. Therefore, by the shape, you could differentiate smooth muscles out, and that leaves you with the skeletal and cardiac muscles. If you take skeletal and cardiac muscles, skeletal muscles are not branched, whereas the cardiac muscles are branched. Again, if you look at the nucleus is located in extreme periphery in the skeletal muscles. In smooth muscles and cardiac muscles, it's centrally located. Then if you go by the striations, smooth muscles have no striations and skeletal muscles and cardiac muscles have striations. Let me ask you one question. Why smooth muscles doesn't have striations?
Because the absence of sarcomeres.
Sorry? Okay, let me ask it in a different way. What causes striations in the skeletal muscle?
That is because of the arrangement of microfilaments, actin and myosin filaments.
Yeah. How arrangement of actin and myosin filament causes striations? Why?
Light band, dark band.
Sorry?
Like actin filaments are thin filaments, myosin filaments are thick filaments. So they are arranged in a manner so that they partially overlap. So they give us a striated appearance like from end to end. Within a sarcomere, they get arranged partially overlapping. So they give us a striated appearance.
What is striated appearance? Let me go back one step. What do you mean by striated appearance?
It's because of sarcomeres.
No. I'm asking simple questions. When you say striated appearance, what does it mean?
Like cross lines.
It looks like alternating light and dark bands, right? There's a one light band, there's a one dark band, one light band, one dark band, due to the arrangement of actin and myosin. That's all. Alternating light and dark bands are known as striations. So that is both present in skeletal and cardiac muscles. If you were asked to describe the light electron microscopic appearance of these striations, yes, you could go and describe this A band, I band, Z band, sarcomeres, all these things. The striations are alternating light and dark bands due to the arrangement of actin and myosin, which is thick and thin filaments, right? So now you can come back and answer my question. Why do smooth muscles lack striations?
They do not have such arrangements of actin and myosin filaments, madam. They are
Yeah, do they have actin and myosin?
yeah, they have,
Yes.
but they are
But
not.
they are not arranged in a alternating light and dark bands. Yes, that's why we call it smooth muscle, because the cytoplasm appears smooth, no alternating light and dark arrangement of actin and myosin fibers, right? Then this T-tubules. Anyone knows about T-tubules? No? You haven't heard about T-tubules? Anyone? What are they?
The sarcolemma being
Yes, near the sarcolemma. What's arranged as T? What is this? From sarcoplasmic reticulum. What is sarcoplasmic reticulum?
the smooth endoplasmic reticulum of skeletal muscles.
Yes,
Skeletal
endoplasmic reticulum of the muscle cell is known as sarcoplasmic reticulum. What is the plasma membrane of muscle cell known as?
sarcolemma.
Sarcolemma. So there's this unique terminology for muscle cells. Endoplasmic reticulum of the muscle cell is known as sarcoplasmic reticulum. So sarcoplasmic reticulum arranged in a peculiar way, forming three tubules. It's known as triads in skeletal muscle and dyads, because there's two tubules are arranged in cardiac muscles. It's known as a dyad. I'll show you a triad and a dyad. Like here. Now you can see that's the sarcolemma, that's the plasma membrane of a skeletal muscle. And then you have this sarcoplasmic reticulum, that is the endoplasmic reticulum of the muscle cells. We call it sarcoplasmic reticulum. And that there's this special tubule known as transverse tubule. In shortened form, we call it a T-tubule here. And then they are here again. and the sarcoplasmic reticulum arranged on either side of this T tubule. So 3 items together. You can see 2 sarcoplasmic reticulum surrounded by a transverse tubule or T tubule. Therefore, there's 3 items. We call it as a triads. We can see this only under ultrastructural level or electron microscopical level. So one way of identifying skeletal muscle is microscopically, that it has this triad arrangement, 3 things together. As opposed to the cardiac muscles, cardiac muscle has dyads, that is 2 things. What are those 2 things? It has this endoplasmic reticulum or sarcoplasmic reticulum, T tubule, and that's it. There's no sarcoplasmic reticulum on the other side. So there's 2, we call it. The cardiac muscle has dyads of 2 tubules. So cardiac dyads, skeletal triads. And the last feature is intercalated disc, and they're absent in everywhere except in cardiac muscle cells. So those are the roughly features that differentiate the 3 types of muscle cells. Right. Okay. Now let's wind up the lecture, and please keep a record of your attendance, right? Okay.
Thank you, madam.
Okay.
Thank you, madam.
Thank you.
Thank you, madam.
Yes.